Cancer is a solvable problem. We have, 3 to 4 billion bases in the genome, and in cancer we get specific mutations that drive the formation of cancer that make them distinct from normal cells. We're focused on developing novel therapeutics for cancer. Specifically, we like to exploit cancer-associated defects and things like DNA repair and cancer metabolism so that we can selectively target cancer over normal tissue. DNA repair is interesting because mutations in DNA repair lead to cancer formation, but they also represent Achilles heels that we can exploit for therapeutic gain. So they're sort of one and the same in terms of they cause cancer, but they also become nodes that we can target for novel therapeutics development. The interplay between DNA repair and cancer metabolism is actually more linked than we had thought of previously. Our laboratory and others have shown that actually cancer metabolism mutations actually induce DNA repair defects, and DNA repair defects can induce changes in cancer metabolism. Yet again, a deeper understanding of how cancers are formed, but also another opportunity to target cancers with novel therapeutics that go after both cancer metabolism and DNA repair. We use a sort of rare combination of very multidisciplinary problem-solving techniques, and this is something that we really leverage in the Yale research community, all the way from synthetic chemistry up in Science Hill to translational cancer modeling here at the Cancer Center and clinical trials. We often will look at basic science, the mechanistic impact of specific mutations, and then we'll use patient-derived tumor samples to do modeling of how those drugs work. And then we'll actually design the clinical trials as a physician scientist treating patients, so we're able to offer insights on how to use that information to guide the development of novel therapeutics trials. One of the biggest problems in our field in radiation oncology is a lot of the therapeutics are universal sensitizers. They sensitize normal tissue, sensitize cancers independent of mutations. So we're pursuing a relatively novel concept, which is tumor-specific radiosensitizers and chemosensitizers as well. And what we're doing is going and looking at those specific mutations, modeling them, and trying to figure out how we could develop novel therapeutics that will only sensitize tumor cells that have those mutations so that the normal tissues can be spared. I've gotten the opportunity to meet some of these families to see how devastating these tumors can be. So professionally, my interaction with patients drives the work in our laboratory. One of the most exciting parts of our research is that we are very translational, and we love to bring things as quickly as possible into the clinic. We made a discovery in 2017 that IDH mutations, cancer metabolism gene, cause DNA repair defects that cause sensitivity to PARP inhibitors, which are FDA approved for BRCA mutant cancers. So we're actually able, by 2017, within 5 years, to run clinical trials in IDH mutant tumors with FDA-approved PARP inhibitors. More recently, we published a paper in Science in 2022 on a new class of chemotherapeutic drugs that target DNA repair mutations in glioblastoma, a malignant brain tumor. The work that we do has direct clinical implications, and that's really what drives us on a daily basis.